Hello, dear students. Today we are going to know about the most important part of any organism. Imagine each of your body parts has its own team of tiny superheroes working tirelessly to keep you alive and functioning. What would their headquarters look like? Dear children, let us dive into the world of amazing tiny structures. Robert Hooke, in the year 1665, observed honeycomb like tiny compartments while examining thin slices of cork. You can see the two pictures given here. The first one is the structure of a plant cell and the second one is the structure of an animal cell. The details about their structures we will be reading in due course of time. Cork. What is cork? It is a substance obtained from the bark of trees. Those tiny compartments were named as cells, which means little rooms in Latin language. Robert Hooke observed dead plant cells. Cell is the basic building block of all living organisms. Like the way we join many bricks to make up a building, many cells come together to form the tissues. Tissues make organs and ultimately the organism. From the smallest bacterium to the largest whale, all living things are composed of one or more cells. That is why we call the cell the fundamental unit of life. The cells are the fundamental and structural units of life. Few cells, such as eggs of ostrich, are the largest and are visible in the naked eye. The smallest cell is the bacterium mycoplasma. Children, please see the three pictures given here. The first one is the example of a bacterium, that is a tiny microscopic organism. The second one is a plant which is macroscopic, and the third one is human, which is also macroscopic. Now, when we take a small section of the onion bulb after carefully peeling off a thin transparent layer, that is epidermis. Epi means open, dermis means skin. So, the layer which lies on the above from the inner layer, and then we add 1 drop of iodine or safranine solution, the structures will be more visible. They are cell wall, cytoplasm and nucleus. You can just view these structures in the given diagram. The cell wall is the outermost layer, then the cytoplasm and then the nucleus as well as the vacuoles. This is the picture of a plant cell, that is why the vacuole is prominently present at the middle and the nucleus is present towards the periphery. In the year 1665, Robert Hooke observed cell in his primitive microscope and published the same in his book Micrographia. In the year 1674, Anton von Leeuwenhoek proposed improved microscope and observed free living cells. In the year 1831, Robert Brown discovered nucleus in the plant cells. In the year 1838, Matthias Ledin concluded that all plants are made up of cells, and in the year 1839, Theodor Schwann proposed that all animals are made up of cells. In the year 1838 and 39, T. Schledin and M. Schwann formulated cell theory. In the year 1855, Rudolf Virchow proposed that cells arise from pre-existing cells. In the year 1940, electron microscope was discovered. The discovery of electron microscope led to the fact that an organism may consist of only one cell or it may consist of many cells. An organism consisting of only one cell is an unicellular organism. The single cell is responsible for carrying out all the life processes, which are also called as metabolic activities, very small in size and can only be visible with a microscope. An organism consisting of only one cell is an unicellular organism. Examples of unicellular organisms are bacteria, amoeba, paramecium, yeast, chlamydomonas, etc. Children, you can see the diagram of bacterium given in this slide, which is an example of an unicellular organism. Now, an organism consisting of many cells, that is more than one cell, is termed as a multicellular organism. Here cell differentiation is there, division of labor is also existing. For example, muscle cells are specialized for movement, nerve cells are specialized for communication. You can see the example of a plant and a human being as the macroscopic organisms or multicellular organisms. They can be visible through naked eye. Many cells combine to form tissues, many tissues combine to form organs, and many organs join to form organ systems, and then ultimately an organism is formed. So, complex organization is there in multicellular organisms. When a group of cells having a common origin and perform a specific function, it is termed as a tissue. Examples of tissues are meristematic tissue, permanent tissue, connective tissue, nervous tissue, epithelial tissue, and muscular tissue. Examples of microscopic multicellular organisms are water bears, rotifers, some nematodes, and microscopic crustaceans. All cells of a tissue work in a coordinated manner. Most are visible through the naked eye. The cell is not just a simple structure, children. It is a miniature world with its own components and functions. Now, let us read what are these different parts inside a cell. We will learn more about these amazing internal structures called organelles and how they help the cell to carry out all the necessary life processes. Now, organelles, these are specialized membrane bound structures within a cell that perform specific functions. Examples are nucleus, mitochondrion, endoplasmic reticulum, etc. You can see the picture. Structure of a nucleus is the first one, structure of mitochondrion is the second one, and structure of rough endoplasmic reticulum is in the third diagram. We will read in detail about all these. Now, inclusions are non-membrane bound structures present in the cytoplasm. They can store nutrients, secretory products, or pigment granules. Examples are glycogen granules, lipid droplets, pigment granules, etc. You can view the picture, children. Starch grain is an example of an inclusion, and alluron grains are also examples of inclusions. Three most important features that are found in almost every cell: protoplasm, cell membrane. These are the two structures. Now, the protoplasm is again divided into two types, that is cytoplasm and nucleoplasm. Children, see the diagram very carefully. The outermost layer is the cell membrane because the structure is of an animal cell, and the nucleus is present at the center. The nucleus is surrounded by nuclear membrane and it consists of cytoplasm and nucleoplasm or karyoplasm. When the cytoplasm and karyoplasm combine with each other, that forms protoplasm, that is the fluid matrix of the cell. Now, protoplasm is also known as the living fluid. Cell membrane, the first structure, is also called as the plasma membrane. It is the vital structure that defines the cell and controls the movement of substances in and out of it. Now, cell membrane is a complex structure which is lipoproteinaceous in nature. Children, remember the word, that is lipoproteinaceous. It means it is made up of lipids and proteins. Here, proteins are embedded inside the phospholipid bilayer. Now, this is the structure of a cell membrane where proteins are present as well as lipid bilayer is existing. Now, cell membrane is selectively permeable. Now, let me discuss three different terms with you right now. The first one is permeable. Permeable means this type of membrane allows the entry and exit of all substances. Impermeable means these type of layers do not allow the entry and exit of any substance. Semi-permeable means they allow the entry and exit of few substances selectively. Thus, they are also called as selectively permeable membranes. Some substances like carbon dioxide and oxygen can move across the cell membrane by the process of diffusion. Now, what is diffusion? Diffusion is the movement of substances, maybe carbon dioxide or oxygen, from a region of high concentration to a region of low concentration. Just focus on the diagram given. Movement of solute from high concentration to low concentration. See, on the left side, the concentration of the molecules is more, that is high solute concentration. In the process of diffusion, they move to the right side, that is less solute concentration. So, diffusion is a process where the movement of substances takes place from high concentration to low concentration. Now, let us learn about osmosis. Osmosis is the movement of water molecules from a region of high concentration to a region of low concentration through a semi-permeable membrane. Osmosis is a specific type of diffusion. Children, see, high concentration to low concentration movement of gases usually is diffusion. When we are talking about osmosis, movement of solvent molecules takes place from high concentration to low concentration through a semi-permeable membrane. Now, osmosis is of two types. One is endoosmosis and the second one is exoosmosis. Endoosmosis is the entry of water into the cell from the surroundings, whereas exoosmosis is the exit of water from the cell to the surroundings. The term endo means in and the term exo means out. Example of endoosmosis is swelling of resins, and example of exoosmosis is shrinkage of skin of fingers in soap water. Now, coming to another term, that is tonicity. Tonicity refers to the relative concentration of solutes, that is dissolved substances in a solution compared to another solution. There are three types of solutions. One is hypotonic solution, second one is hypertonic solution, and the third one is isotonic solution. Hypotonic solution has low concentration of solutes outside than inside the cell. Therefore, water enters into the cell and the cell swells, or we can say the cell becomes turgid. See the picture. If a cell is kept in a hypotonic solution, then movement of water takes place into the cell and the cell swells. Now, this means the concentration of water is more, that is the concentration of solvent is more outside than inside the cell. So, water enters into the cell or the cell becomes turgid. Now, hypertonic solution. This has high concentration of solutes outside the cell than inside. Therefore, water moves from the cell to the external surrounding and the cell shrinks, or else we can say that the cell is plasmolyzed, and the entire process of shrinkage of cell is called plasmolysis. Students, see the picture. If a cell is kept in a hypertonic solution, then water moves from the cell to the surrounding. Therefore, the size of the cell decreases or the cell shrinks. Moving on to the next picture, when we are talking about a plant cell and a plasmolyzed plant cell. Here, the cell wall remains intact because that is rigid and impermeable, whereas the cell membrane shrinks, and this process is called plasmolysis. The water concentration outside is less than inside the cell. Thus, water moves out of the cell and the cell shrinks. The cell is plasmolyzed. Cytoplasm shrinks and pulls away from the cell wall. The cell wall remains intact but the cell membrane contracts. This process is called plasmolysis. Moving on to the third type of solution, that is isotonic solution. Iso means similar. So here the concentration of solutes outside and inside the cell remains same. Children, see the diagram. If a cell is kept in an isotonic solution, the cell size remains same. No change takes place because the concentration of both inside and outside the cell of the solutes is same. Dear students, let us know more about solution. When solute combines with solvent, solution is formed. And when we say the solute concentration is more, that means concentration of solvent is less and the concentration of solution is more. When we say less concentration of solute, that means solvent concentration is more and concentration of solution is less. The flexibility of the cell membrane also enables the cell to engulf food and other materials from the external environment. For example, amoeba engulfs food or other substances by surrounding the food particle through pseudopodia or false feet and forms a large vesicle called phagosome. The process by which cell engulfs substances from outside environment is called endocytosis. Engulfing solid particles is called phagocytosis. Engulfing liquid droplets is called pinocytosis. You can see the diagrams. First one is the diagram of amoeba. Second one depicts the picture of phagocytosis, that is how the pseudopodia or the false foot of amoeba surrounds the food particle and then automatically dissolves into the cytoplasm of amoeba. And the third one is pinocytosis, where liquid droplets come closer to the cell membrane of amoeba, then gradually they dissolve into the cytoplasm. So, phagocytosis is also known as cell eating and pinocytosis is also known as cell drinking. Now, moving on to the other structure, that is cell wall. This provides structural support and protection to the cell. Chemically, cell wall is mainly made up of cellulose. It may contain hemicellulose, pectin, lignin, chitin as in plants or peptidoglycan as in bacteria. Now, cell wall is basically made up of cellulose. You can see in the diagram. Here, inner layer is there, middle layer is there, outer layer is there, primary cell wall and secondary cell wall. Now, the function of cell wall is it provides structural support and maintains the cell shape. It protects the cell from external injury. It regulates the cell growth. It controls the movement of molecules and it provides rigidity. A membrane-bound organelle that contains the genetic material of the cell is the nucleus. The nucleus has a double-layered covering called nuclear envelope or nuclear membrane. The nuclear membrane has pores which are called nuclear pores and that regulate the movement of molecules between the nucleus and the cytoplasm. The nucleus contains thread-like rod-shaped structures made up of DNA and proteins. These are responsible for carrying the genetic material. Children, see the structure of nucleus. It consists of outer membrane, inner membrane. In between the outer and inner membrane, there is a space which is called as the intermembranous space, and these two consist of the nuclear envelope. Inside the nuclear envelope, there is a sap or fluid present which is called as the nucleoplasm. And this nucleoplasm consists of chromatin threads, that is DNA and protein structures. They are found floating inside the nucleoplasm. And then ribosomes are also attached to the outer membrane sometimes. And this nuclear membrane, nucleus, all these help in storing the genetic material. That means the function is it stores genetic material, it protects the DNA from damage. This is the site of DNA replication, and nucleolus, that is the dense structure which is the primary site of ribosomal RNA synthesis. Students, After knowing about the detailed structure of nucleus, let us learn about two types of cells, prokaryotic cells and eukaryotic cells. Prokaryotic, pro what means primitive and karyon means nucleus. Eu means good or advanced and karyon means nucleus. So when we are talking about prokaryotic cells, definitely these are primitive, simple and small cells, whereas eukaryotic cells are more complex and large in nature. See the structure of two diagrams given here. The first diagram depicts the cell of a bacterium which is prokaryotic in nature, whereas the second one is the structure of an animal cell which is eukaryotic in nature. See, in the first diagram, there is no confined nucleus present at the center of the cell because there is no demarcation line for the nucleus. There is no differentiation, and the chromosomal material, they are present at the center in the form of nucleoid. Whereas in the second diagram, you can see there is a clearly differentiated nuclear membrane and nucleus. So, cytoplasm is completely differentiated from the karyoplasm. Now, what is cytoplasm? These are gel-like substances that fills the region between the cell membrane and the nucleus. This consists of cytosol and organelles. What is cytosol then? This is the fluid portion which is a complex mixture of water, ions and macromolecules, proteins, carbohydrates, lipids as well as nucleic acid. Organelles are membrane-bound structures as we have already discussed. Those are found suspended within the cytosol, each with specialized functions. And there is another term, that is cytoskeletons. These are a network of protein fibers that provide structural support and shape to the cell. What is the function of cytoplasm? This supports the cellular structure. This helps the suspension of organelles and inclusions inside the cell. This is the site of metabolic activities inside the cell. Metabolic activities means life processes that are taking place inside the cell. This helps in the storage of molecules as well as this helps in the transport of materials. Now, moving on to the next organelle of a cell, that is endoplasmic reticulum. This is an extensive network of membranes that extends throughout the cytoplasm of eukaryotic cells. This plays a crucial role in protein and lipid synthesis. Let us learn more about endoplasmic reticulum. This consists of a network of interconnected sacs and tubules called cisternae. Two main types of endoplasmic reticulums are there. One is smooth endoplasmic reticulum and the second one is rough endoplasmic reticulum. Now, first one is SER, which lack ribosomes, and the second one is RER, which contains ribosomes. That is why they look rough. Now, what are the functions of endoplasmic reticulum? Endoplasmic reticulum, specifically rough one, helps in protein synthesis because ribosomes are found attached with this type of endoplasmic reticulum. And you can see the same thing in the second diagram. That is the diagram of a rough endoplasmic reticulum where dots are found in the wall of the endoplasmic reticulum. Those dots represent the ribosomes. Whereas smooth endoplasmic reticulum or SER helps in lipid synthesis and detoxification as well as calcium storage. And rough endoplasmic reticulum as well as smooth endoplasmic reticulum both help in protein folding and modification. This also helps in membrane synthesis as well as formation of transport vesicles. Moving on to the next organelle of a cell, that is Golgi apparatus. This is also called as Golgi body. You can see the picture of Camillo Golgi. He was the first scientist who observed Golgi apparatus inside the microscope. This is composed of a series of flattened membrane bound sacs called cisternae, and the functions are: number 1, it helps in the processing of protein and modification. This also helps in lipid processing. This also helps in storage and packaging as well as vesicle formation. Protein and lipids are transported to their final destinations with the help of Golgi complex. This also helps in secretion of substances and formation of lysosomes. Now students, we will know about the details of lysosomes. These are membrane bound sacs filled with digestive enzymes. These are a kind of waste disposal system in cells. You can say all the waste that are gathered from all the organelles inside a cell can be disposed of because of the presence of lysosomes. And its function is it keeps the cell clean by digesting any foreign or worn out cell organelle. These contain powerful digestive enzymes which are capable of breaking down all organic material. That is why these are also known as the suicide bags of cells. Children, you can see the diagram of the lysosome which consists of a number of enzymes, and almost all the enzymes are hydrolytic in nature because hydrolytic enzymes use water. The term hydro itself indicates water. So, these enzymes use water to break down larger molecules into smaller ones. The general reaction catalyzed by hydrolytic enzymes helps in the disposal of waste from the cells. Now, moving on to the next most important organelle of a cell, that is mitochondria. This is also known as the powerhouse of the cell. This can be oval, rod-shaped or capsule-shaped organelles. Double membranous structure. Outer membrane is smooth. Inner membrane is folded into cristae, thus increases the surface area. Cristae encloses mitochondrial matrix. Matrix is the fluid substance that is present inside the mitochondrion. There is intermembranous space between the outer and the inner membrane. Matrix is the space enclosed by the inner membrane. They have their own DNA. Let us see the detailed structure of mitochondrion. This is called as the powerhouse of the cell. Just see the structure. This consists of outer smooth, inner folded membrane, inside matrix is present, and then intermembranous space is present. And the functions of this mitochondrion is, this is the site of cellular respiration. What is respiration, children? This is the process that converts the nutrients into energy in the form of ATP. And this is the only place where energy is produced, and that is why the most important organelle of all the cells. This also helps in glycolysis. What is glycolysis then? This is the process of breaking down of glucose into pyruvate. Now, oxidation is also taking place here. The pyruvate which is formed is again converted into acetyl coenzyme A, and then oxidative phosphorylation takes place, because of which ATP is generated. What is the full form of ATP, dear students? It is adenosine triphosphate. Dear students, now we came to know that different cells are responsible for different functions. Nerve cells help in communication, and root hair cells are adapted for absorption. Cells are the fundamental unit of life. They are the structural and the functional unit of life. Now students, this is all about today's class. In the next class, we will be discussing about how a single cell divides to form a group of cells, that is tissues, types of plant and animal tissues, along with their structure and functions. Till then, bye-bye.